So let's continue with the muscle tissue. And this is about the coverings of the muscle, the connective tissue, dense, irregular, epimysium. Epimysium is the outermost. So that pretty much covers the muscle bellies, which you're going to see. And we'll start muscle after the fourth, I believe. We'll start getting into naming the muscles and all that. And you can see the outer portion of the muscle is epimysium, dense, irregular connective tissue. So that's not muscle, it's not conductive, it's just covering. And then we have these things called fascicles, which are groups of muscle cells. Sometimes we say muscle cells is the same as muscle fibers. So the perimysium surrounds these fascicles, and within the fascicles, you have muscle fibers. And that's the same thing as a cell, a muscle cell, which we're going to get deep into. And the endomysium is kind of between each muscle fiber. Is it that important to me? Not really, because I'm more concerned about the membrane of the muscle cell and the fluid around there. So this all is just packaging, pretty straightforward. And this is a really good picture. So you might actually see this on that third practical where you just have to name the coverings. Like you can see the bone, you can see this is the femur here. And this must be a quadricep tendon or origin of the quadricep. So all these muscle bellies really are covered by epimysium. And then you have these fascicles all wrapped up in perimysium. So all these are fascicles. Like this is a fascicle. And each one of these little tubes is a muscle fiber. So these cells are very irregular. They look like little tubes, and they have these really small compartments, which we'll talk about. And that's really where the magic happens. So this is just gross anatomy, things you could see with the naked eye, right? You really can't see it when you look at the models or the pictures that we're going to look at, but you can see what is inside the muscles as you go deeper. But know like where the tendon is, like by the greater trochanter and then the lesser trochanter, the femur, just to get some idea of where you are. Here's the shaft of the femur. And we'll take it from there. So the attachments, and you know this, right? You know that a tendon attaches muscle to bone, and that's tendon is dense, regular connective tissue. So that connects muscle to bone. Of course, I'll ask you that. And ligaments attach muscle to muscle. Also dense, regular connective tissue. So bone to bone. And then later we'll see what's called fascia or aponeurosis. Will kind of be in between the muscles, connective tissue called fascia. That's just a line here I drew. So fascia or aponeurosis, basically the same thing. And usually that's dense, irregular connective tissue. So this is important to get these names down, the attachment. So the insertion is the movable part. So it's on the bone that's going to be moving. Like for the quadriceps muscle to extend the knee, the insertion has to be down on the tibia. And we'll look at the tibia tomorrow and some of the bony markings on the tibia. The origin is pretty much where the muscle starts, and it's not the movable part, like up on the femur, on the proximal femur. So where the quadriceps actually originate from, and that's the immovable. All right, so that's extension of the knee and the quadriceps, bringing your toes up, or bringing your whole leg up, I should say. So you can have indirect or direct attachments, like direct or right onto the epimysium. Indirect is through your tendons or aponeurosis, which is also known as fascia. So we'll see, and you'll be able to name some of the aponeuroses, and you can see where the tendons are in the muscles, and mostly in lab. So again, that's the same thing. And you look at different descriptions of muscles. Okay, you can see this. I always like these tables because they kind of organize everything. So you can see the epimysium, the perimysium now is getting deeper, and then the endomysium, which is between the muscle fibers. So this is an actual muscle cell or muscle fiber. So this is really where I get interested in this, because you have below the endomysium, you have the membrane. And the membrane of a muscle is called the sarcolemma. Sarcolemma is another name, very important. Sarco means muscle. And within the muscle cell, you have these myofibrils. And I might have mentioned last week the proteins, the myofilaments that make up myofibrils, like actin and myosin. There's no connective tissue around those. So these are the contractile proteins that are going to move and shorten the muscle, because really that's really all the muscle does. It contracts by shortening. So you're going to learn this part along these myofibrils called the sarcomere. So right now, this is really the important thing, right? You know the difference between the epimysium, perimysium, and endomysium, but really we want to get into the actual membrane of the muscle and what's inside that membrane, like cytoplasm and calcium channels, and really important when we get into that. So the sarcolemma, remember sarco means muscle, is the very excitable plasma membrane. And sarcoplasm is the cytoplasm, what's inside the cell, which is organelles, just like anything. Like you're going to have nucleus, you're going to have ribosomes, right? You're going to have areas for glycogen storage. Remember, skeletal muscle we're talking about here. Right? Skeletal muscle is where glycogen is stored, and the storage area for glycogen is called glycosomes. So this is important. Really, you should just know glycogen is stored there, because I like this word too. Myoglobin is a place for oxygen storage, which is really unique to muscle cells, especially skeletal and cardiac. So skeletal muscle cells within the sarcoplasm store glycogen and they store oxygen. So this means we're going to have to make a lot of ATP, right? For muscle contraction, you need ATP. So that's your main ingredients for making ATP to make the muscle contraction happen. So myofibrils, you're going to see, are the actin and myosin, and there's a couple of other proteins. And this is another really important organelle, because the sarcoplasmic reticulum is all about storing and releasing calcium. Why do we study all that calcium in the bone? And this is why. Really important, because we're going to need it for not only muscle contraction, but also for nerve conduction as well. And in a skeletal muscle, you can't contract a muscle without a nerve impulse. In fact, every muscle needs a nerve impulse to contract. Maybe you'll have an argument with smooth muscle, but we're only talking about skeletal muscle this semester. It's all about voluntary contraction, where you use your brain to contract the muscle. So through the nervous system, of course. So the muscle needs the nervous system. So the nervous system needs calcium just to get that signal. And then the muscle needs the calcium to contract. Can't happen without that, right? We'll talk about T-tubules, how that connects the channels for calcium within the sarcoplasm. All right, let me just mute these mics. Yeah, that's not fair. Okay. So let's move on. So in the myofibrils, now these are within the muscle cell or within the muscle fiber. So, and these are not the striations you can see on the outer part of the muscle, but striations are microscopic. And striations are alternating proteins, like I'll say actin is light colored and it's thin. And I'll show you what that looks like in a bit. And myosin is kind of the opposite, is dark and it's thick. Okay. So sarcomeres are basically the smallest unit of a muscle fiber, small as you can go. And they're basically they're structurally described, and you'll see what this means in a bit. Z disc to Z disc, and that's the structure of it. That's important because that's kind of the way it's described. And of course, everything's in between, like actin and myosin, and other bands and lines. The myofilaments are the actin and myosin, right? They are the actin and myosin, which causes the striations. And they're contractile proteins. They cause the contraction ultimately. And they're proteins, which have to be transcribed from your DNA and then translated out on the ribosome with messenger RNA. So we'll look at the component of the composition and what it looks like in the other proteins, like regulatory proteins. So right now, be familiar with actin and myosin. I might have mentioned the other ones last time just to give you the vocabulary. Okay, so again, this is microscopic. You can't see this with the naked eye, not gross anatomy. And you can see that this is a nucleus of a myocyte, skeletal muscle cell or muscle fiber. And if you remember, one of the unique things about skeletal muscle is it's multinucleated, and the nucleus is peripheral, more towards the outside of the cell. Okay, so here's the sarcolemma. We're not talking about endomysium anymore. And of course, and I should mention this, because you're making so much ATP in the presence of oxygen, you're going to need a lot of mitochondria. So now these are all the parts that are really important. So here you can see dark band, which is going to be called an A band. I remember the A band is dark because there's an A in dark. And the light band has an I in it. So I remember that's the light band. So you're going to see when we get into the sarcomeres, these are many, 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 many sarcomeres kind of lined up together along each one of these little myofibrils. So get the term down, myofibrils. We're not in fascicles anymore. We're way deep inside the muscle cell, which is really important. And there's fluid in there too. You have intracellular fluid and many ions through those T tubules and sarcoplasmic reticulum, mitochondria, all of that, all the organelles. So this really, it's great that I can explain it, but we have to look at this. You know, the A bands are the dark regions. The I bands are the light. The Z discs are right in the middle of the I bands. So like I said before, the sarcomere is anatomically defined, geographically defined from Z disc to Z disc. Okay, I don't care if you put a K or a C. Doesn't matter. The M line is dead center sarcomere. So you kind of figure that one out when you see it. An H zone surrounds the M line. So you will get pictures, and you'll probably see this in your practice questions of what that looks like. Again, this is more gross microscopic anatomy, sorry, but just a look at, these are two different fibers, right? Here's one fiber, here's another fiber in the beginning of the next fiber. And you can see these alternating bands or striations, dark fibers to light fibers. So once we get in deeper, you can see. And we really just have to take a look. So here you go. This is along a myofibril, and you have the filaments. So a sarcomere is between Z disc to Z disc. That's the sarcomere. Everything in between. The H zone is this right here, and it's kind of a light area. This dark part is the M line, and you can see the Z disc is right in the middle of both side I bands. And all this in here, between these two lines here, is your A band. So when a muscle contracts, there's millions of these. I could be exaggerating, but it's a lot. Many sarcomeres along one muscle group. It depends on the size of the muscle, the group of muscles. But what's going to happen is when a muscle contracts, all the Z discs are going to move closer. So all the actin is going to move towards the middle of the sarcomere, and this whole sarcomere is going to shorten. But picture that thousands of times over. And that's really the microscopic, the sliding filament theory of muscle contraction. So there's a lot of steps that have to happen. And remember, this is voluntary, where my cerebral cortex, the motor portion of my cerebral cortex, the primary motor area for somatic, remember this is somatic, not autonomic, somatic muscle contraction, voluntary muscle contraction of skeletal muscle. So the effector is skeletal muscle for this part of the nervous system, the somatic nervous system. I know I keep saying this, but it's really important. So the somatic motor neuron, that's a nerve cell for the nervous system, is going to stimulate this contraction where the Z discs are going to get closer over and over and over. And they, of course, they get excited when this happens. You get excitement, depolarization, then contraction. And then they have to relax too. They can't stay contracted. That would be completely inappropriate, except for some muscles that are postural muscles that need to stay contracted to hold you upright. But usually one muscle contraction has to repolarize and relax, and you'll see what all that means. Okay, the myofilaments are your proteins, your contractile proteins, your actin and myosin. Remember, actin's thin. It's kind of, a bead-like protein, like this across, and it has binding sites on it for myosin. So myosin ultimately is going to bind to actin. So these little circles I'm drawing are the binding sites for myosin on actin. And myosin looks more like a thick, kind of like a golf club configuration, and it's dark and thicker than actin. So myosin is going to bind to that actin binding site. It's going to swing the actin towards the middle of the sarcomere. Remember, sarcomere Z disc to Z disc. The M line is dead center. And most of the A band is made up of dark fibers. Remember, A and dark for dark fibers. And the dark fibers are mostly the myofilaments called myosin. The light ones in the I band, probably more actin, right? Thin, light. I band, there's an I in light. There's an A in dark. And the M line is dead center. So myosin is swinging the actin. Myosin is connected to the M line, which you'll see. And the actin, and this happens on two sides too, which you'll see. Like this is the, right here, the Z disc is here. Well, there'll be Z disc on the other side. And that actin will come towards the middle. And of course, that's what contraction is. The I bands come, the Z discs come together. The I bands pretty much stay where they are. The H zone disappears, which I'll show you that. So here's like a cartoon of this all, one sarcomere. And you can see in here, you have the I band around the Z disc. The Z disc is in the middle of the I band. So this is one side, and this is the other. So, and this repeats itself palindromically over and over and over. And you have different proteins too. You'll see different proteins, like right here, this protein, which I'll show you later, adds support and some elasticity to the sarcomere. So right here, this is all myosin, all this right here. And then this is your actin on both sides. M line is dead center. H zone is here. So this would be your H zone. And that's basically going to disappear. Like in a maximally contracted sarcomere, the H zone basically disappears as the Z discs come closer together, swinging the actin towards the middle of the sarcomere. So this is kind of confusing down here, this cross-section business. I would never ask you in that position, but it's, once you read this and you go through, you could see the thicker filaments in the H zone and the this is all like A band, right? I band is more thin filaments, like your actin. So get those down. So again, parts of the myosin, like I think we're talking mostly about myosin here. right, with heavy chains. And we're just worried about the heavy part. And the tail is basically the golf club shaft, right? This part is the tail. And then it has like a globular head. So this would be the head up here. And that's the part that's going to bind to actin. So when myosin binds to actin, that's called a cross bridge. It's called a cross bridge between myosin and actin. Okay. Thin filaments, again, globular proteins, thinner, right? You don't have to worry so much about F-actin or G-actin, but you do have to worry about one thing. And on actin, there's a binding site for myosin. So this, if this is one of the proteins of actin, right, it has a binding site where myosin needs to bind. Now, here's the problem. In a relaxed muscle, before it's stimulated, you have two proteins. One protein covers the binding site for myosin on actin, and that's tropomyosin. Troponin is attached to the tropomyosin on top. So what has to happen? First of all, the nerve membrane, the muscle membrane has to be excited by a motor neuron. And then there's a whole chain of events that happens. So what I'm telling you now is the sliding filament theory, the actual contraction of the sarcomeres. But there's a lot of steps that have to build up to this. So understand this. Before this could happen, you have to think about moving a muscle because it's voluntary. And that comes from an upper motor neuron from your cerebral cortex. And eventually it crosses over, actually. So if I want to move my right hand to right, the left side of my cerebral cortex in the primary motor area starts the signal. So that signal crosses over, neurons cross over to the right. And then ultimately, the lower motor neuron will get to the muscle that I want to move, like my hand or my arm. And that all has to happen through excitation, depolarization, action potential, all these words that have to do with the nerve cell. And then when it gets to the muscle membrane, the sarcolemma, then that membrane has to be excited, depolarized, reach an action potential. And when that happens, there's this ion, it's called calcium, which I mentioned. Calcium, remember this, calcium is released from the sarcoplasmic reticulum and it binds to troponin. So once calcium binds to troponin, it moves tropomyosin off of the actin binding site for myosin. So now myosin can bind. And none of this can happen without calcium being present in the sarcoplasm. And where do we get calcium from? It's stored in the sarcoplasmic reticulum. So remember this. Troponin is what binds calcium. Tropomyosin is blocking the binding sites on actin for myosin. So once calcium binds to troponin, the tropomyosin can be displaced and the myosin can bind to actin and then form a cross bridge, right? Remember that word, cross bridge. Okay. So tropomyosin and troponin are regulatory proteins. Technically, they're not contractile proteins. They're regulatory proteins. Okay, so this is showing the thick filament in the myosin heads, those little golf club heads. Like here's just one strand. All right, on top of there. So this is the binding sites for actin on myosin, but myosin is going to bind to actin. All right, what just happened here? Let me get rid of this. So those are really important points here. You know, the binding of these two proteins, right? X really important. So know the two proteins, the actin and myosin, and then troponin, tropomyosin. So this is what your book really doesn't tell you. So pay attention to this. First of all, in order for this to move, this myosin head to move and find actin, the binding site on actin from myosin, ATP has to be hydrolyzed. So ATP is present. That's why we need ATP. And it's hydrolyzed to ADP plus a phosphate. And the phosphate's really what energizes this myosin head. So you need ATP hydrolysis. This is called hydrolysis, where you add a water to cleave the ATP. And that's where the energy comes from. It comes from that free phosphate, right? Adenosine triphosphate will give you adenosine diphosphate, 2 phosphates plus a free phosphate. And that energizes the myosin head. So it has to move to actually form that cross bridge with myosin, with actin. So here's actin in its resting form, and you can see those little circles that are binding sites for myosin. And here's your tropomyosin, is that long strand-like protein that's blocking the binding sites. And attached to the tropomyosin is the troponin. And troponin is where that calcium is going to bind. Okay, so you can see the binding sites, and they're covered with tropomyosin. So without calcium, this could never happen, and you could never shorten a sarcomere without the homeostatic calcium. So there are other proteins that are involved. One is titin. Sorry, I just had a sneeze. And titin is that string-like, stretchy, recoil-looking protein. Dystrophin also links the filaments to, it keeps the integrity of the sarcomere, really. Like there's a condition called MD or DMD, Duchenne's muscular dystrophy, is a problem with the machinery that makes dystrophin, the protein. So there's a mutation, a sex-linked mutation genetically, where dystrophin is not made correctly. And if without having that strength in the sarcomere, you'll have infiltration of fatty tissue and you won't get good muscle contraction. So a genetic problem in making dystrophin leads to a condition called Duchenne's muscular dystrophy, which is very weak legs, barely able to walk. And it affects boys more because it's sex-linked, because females have 2 X chromosomes where males just have an X and a Y. So it's really hard to cover up that recessive gene in males. So muscular dystrophy is more common in boys, and especially young boys.
My cousin has muscular dystrophy.
Not a good thing. So I'm sorry to hear that.
He can barely he can't barely move his arms too.
Yeah, that's not a good thing. Not a good thing. It's depressing to talk about, Christina. But what can you do? All right, Natalie? Natalia, sorry, was that you, Natalia?
Yes.
Unfortunately to hear that, yeah. Okay, we'll talk about something better later. Okay, so again, that's the link between the sarcomere and the protein. So it gives it strength. And there's other proteins. Nebulin's a good one to remember. Also structural, myomycin, the C proteins. But let's stick to the titin, the dystrophin, and the nebulin, and then actin-myosin, and troponin and tropomyosin. A lot of proteins that we're learning here. So again, the integrity of the sarcomere is very important to get a good muscle contraction. And muscle contraction gets work done. It's not just to release heat and burn up calories and give off water. It's about doing work, skeletal muscles. Oh, here you go. Duchenne's muscular dystrophy. Yeah. Okay, we talked about that already. I don't want to go back there. So it's a defective gene. It's a mutation, autosomal, sex-linked recessive, mostly in boys, unfortunately. And it's usually infiltrated by fat cells because the muscle cells are basically dying. So there's a lot of calcium coming into the cell, but it does no good. So it's destructive and toxic. I've seen this picture before. I don't really want to look at that anymore. Okay. So it's all, supportive care. Physiotherapy could be supportive. There's no exact cure for a genetic condition like this. It's really bad luck. Roll of the dice. Okay, so let's snap out of that and talk about more sarcoplasmic reticulum. Sometimes I just call it the SR. So what are we doing here? This is it. Stores and releases calcium, but it's not going to release the calcium unless there's an action potential in the muscle. We're going to talk about a lot about action potential between now and the end of the semester. So action potential is electric. Major depolarization. You need heavy depolarization above a threshold. And you're going to see what that means. So action potential stimulates the muscle to contract. And action potential is in the neuron as well. We're going to see a lot of this, so we'll take a look. So sarcoplasmic reticulum has tubules which carry calcium through the muscle cell. It's unlike any other cell you've ever seen or ever going to see. The muscle cell is completely unique, the way the organelles are set up and the way the calcium channels are set up as well. So the T tubules, again, they increase the surface area for contraction, which you'll see. And again, I'm not going to ask you about the triad too much because I'm really concerned about the sarcoplasmic reticulum. And it's all about releasing calcium, right? And it forms a network of calcium channels. And that's what all these tubules in the triad, which is the terminal cistern and the T-tubule and the other exiting cistern. So it's all about storing calcium and releasing calcium just so it can bind to troponin to displace the tropomyosin and allow the myosin head, which is energized by the hydrolysis of ATP, to form a cross bridge with actin. And that's as far as we got so far. And there's more to it. It doesn't stop. All right. I'm not going to talk about the triads too much because let's look at it. I think that's the best way to learn this. All right. And now you should be pretty comfortable with the sarcomere. Here's your Z disc. The Z disc, know where the I bands are, light, right? I and light. There's your mid-dead center sarcomeres, your M line, surrounded by that H zone. And the H zone is going to disappear in a maximally contracted sarcomere. Look at all the mitochondria, right? We got to make a lot of ATP for this son of a gun. The myofibrils are within each fiber, and that's the proteins, the actin myosin, which are myofilaments. And the tubules, look at the way they surround the muscle cells. And you have different ends and patterns, and the triad is basically just all this blue, carrying ions, mostly calcium, right, to make sure it can get to every part of that sarcomere. And that's really what the T-tube is all about. They're canals or tubes for calcium to maneuver around the muscle cell. Crazy, man, I'm telling you. Anybody have any questions about that? I'll go to the next one really quick. And we'll talk more about muscle contraction. And we really have to put this together, because this is pretty much what's happening in the sarcomere. But like I said, there's other things that have to happen before and then after in a muscle contraction. So again, the contraction is getting the Z disc to come together and shortening the sarcomere. And the cross bridge is between actin and myosin. So, of course, that has to stop. Contraction can't go on because that would be inappropriate, unless it's a constant stimulus like postural muscles or something like that. So a contraction stopping, a contraction ending, it happens for a couple of reasons. And in the sarcomere, it happens because there's detachment. I think that's 2 words. Detachment of myosin from actin or actin from myosin. So actin comes off of myosin, so that's detachment. And this needs more ATP. to have that happen. So ATP is extremely important. Oxygen is extremely important. That's why we store oxygen in the form of myoglobin. And to make ATP, you're going to need glucose. That's why we store glucose as glycogen in those glycosomes. So the cross bridge begins the connection. But so far, let me just map this out for you. Technically, the first step of the sliding filament theory is hydrolysis of ATP. To energize the myosin head. That has to happen. So this is all happening within the sarcomere. Number two, you're going to form a cross bridge between the two, between actin and myosin. Number three, you're going to swing or bring the actin on both sides of the sarcomere towards the center. And that's called the power stroke. And the last step is detachment of actin from myosin. Why did I put that extra T? I don't know. Okay. So these are the 4 steps that happen within the sarcomere. Now we have to get to that step though. You have to get here. And how does that happen? First is your cerebral cortex. Has to think about it. My spelling is really bad today. Every day. So the cerebral cortex is your voluntary thought. And then you're going to need a motor neuron. And all of this has to be excited through action potential. So there's an action potential of the motor neuron. And the motor neuron is going to excite the muscle, or the muscle membrane, or the sarcolemma. And that's going to have an action potential. And once you have an action potential in the muscle membrane, what's going to happen? You're going to release calcium from the sarcoplasmic reticulum. Now, once the calcium is released from the sarcoplasmic reticulum, what protein is it going to bind to? Do you remember? Is it troponin or tropomyosin? Nobody remembers? That's a really important question. It's troponin. So calcium is going to bind to troponin. It's going to displace the tropomyosin off the binding sites for actin. And then you start the contraction, which is back to step 1. Hydrolysis of ATP on the myosin head to energize it. The second step is the connection between actin and myosin, which is a cross bridge. The third step is moving the actin towards the center of the sarcomere, towards the M line, Z discs coming closer together. And the final step is detachment, which needs more ATP to stop the muscle contraction. And the calcium at the end of the contraction will go back into the SR, back into the sarcoplasmic reticulum to be stored for the next muscle contraction, the next beginning. So just to repeat, this is complicated. You know, it's simple, but it's complicated when you're throwing so many different steps at you. So this is skeletal muscle contraction. So skeletal muscle is voluntary. So you can think about it through your brain to start this contraction. So the brain neuron is going to excite the motor neuron, which is a nerve cell. And that excitement is an action potential. And then the motor neuron is going to excite the muscle membrane. And the muscle is going to have an action potential. And once the muscle has an action potential, all this can start. Releasing calcium from the sarcoplasmic reticulum, calcium binding to troponin. Troponin binding to calcium will displace the tropomyosin off of the binding sites for myosin on actin. And as this is happening, you have hydrolysis of ATP due to this action potential, energizing the myosin head to move into position to form a cross bridge with actin. And then that free energy that you got from the hydrolysis of ATP will further allow for this movement, a strong power stroke, flinging the actin towards the center of the sarcomere. And that's the contraction, that's the shortening. And this can't stay in that position, right? It has to be able to be detached. And that takes ATP too. So this is all the basic flow of voluntary muscle contraction. Now, you're going to learn about the neuron. We have to go back to the neuron and talk about the neurotransmitter called acetylcholine. That's important for this, the chemical part of that contraction or that nerve excitation of the muscle. So all of this was that sliding, the hydrolysis, the actin binding with myosin is all that sliding filament model of contraction. So it's really important to know those 4 steps from hydrolysis to detachment and the cross bridges or the connection. I know this is kind of repetitive, but it should be, right? So what happens in the shortening and that's the contraction to do the work. Shortening of the whole muscle fiber from sarcomere to sarcomere to sarcomere, they're very repetitive. So the Z discs are pulled towards the middle, towards each other, and the M line is dead center. The I bands get a little shorter, and the Z discs, of course, become closer, and the H zone, which surrounds the M line, disappears in a maximally contracted muscle. You could have partially contracted muscles as well. And of course, the A bands move closer to each other because everything's going towards the middle of the sarcomere. So, looking at a fully relaxed sarcomere, right? And you can see this dark Z disc, right? On one side, Z disc on the other. This is all I band, light, right? I and light. This is all A band here. All this is A band. Your M line in the middle and your H zone. So this is all your dark fibers, your thick fibers. And actin you can't really see so well because it's lighter. I'm sorry, this is titin. This is titin here. And this is your actin on both sides. Here's your myosin heads on both sides. So ultimately, the Z disc is going to move closer. The H zone is going to disappear. And the I bands get shorter, smaller, and the A bands come together, as do the Z discs. So this is a fully contracted muscle now. So again, look at the one before. Look at the one there. So everything, the actin is slung towards the middle. So the H zone disappeared, no H zone. Right? The I band just gets a little smaller. And this happens over and over along the length of a muscle fiber, a muscle cell, same thing. Okay, so here's how it goes. Here's what I was trying to describe. So it all starts with the decision to move a muscle, or to move anything. Move your body, move your leg, move your arm, move your head. So your brain, but specifically in your brain, it's the primary motor cortex of the cerebrum. So you could just say cerebral cortex if you like for now. The thing is, you're going to be doing neuro later on this semester, and you have to know exactly what that's called. And you'll see it's called the precentral gyrus of the frontal lobe, which is your primary motor cortex or primary motor area of your brain. So this is all voluntary volition. And a signal is transmitted, right? And a signal is depolarization, action potential, down to the spinal cord, which is below the brain, on the way out to the motor neurons. Now, these are all nerve cells. And then they activate by causing an action potential in the muscle fibers from the sarcolemma is excitable. Right? So this is a I'm going to call this right now, I'm just going to say neuromuscular junction. So without the nervous system, the neuron exciting the muscle, you're not going to get a muscle contraction. So excitable cells changing. Now you're going to hear this word called resting membrane potential voltage. All right, because once you reverse that membrane potential, you get depolarization. And depolarization is going to lead to action potential. And it's all about sodium and potassium at this point when we talk about depolarization. So we're going to go a little further in a bit, and we're going to talk about the chemical part of the neuromuscular junction, and that's a neurotransmitter, excitatory neurotransmitter called acetylcholine. But before we go to that, and that'll be probably the last thing, but you have to understand what depolarization means. And let me start this way. Resting membrane potential, and this is on neuron membranes and muscle membrane sarcolemas. Resting membrane potential is very polarized, meaning that the inside is very negatively charged. The inside of the membrane is negative and the outside is very positive. So the resting membrane potential is, let's say, minus 70 or 90, it could be, millivolts. It's very negative either way, because the neuron is usually about minus 70, but let's just put them together. Any membrane, muscle or nerve, neuron or muscle fiber. So this means it's very polarized. It's very negative on the inside, minus 70 millivolts. Now, sodium is a positive ion, which I hope I got through to you at the beginning of the semester, is mostly on the outside of the cell. So sodium makes the outside of the cell very positive. So when sodium enters the cell, like they call it influx into the cell, because there was a stimulus, like a thought, even on a neuron. That's called depolarization. So now this negative membrane potential of minus 70 inside the cell will start to get more positive. So if it reaches, say, minus 55, which is less negative millivolts, that is a threshold for action potential. Don't worry, I'll tell you. Action potential. So an action potential is usually reaches a potential of plus 30 or plus 40 even, you'll see. Plus now, way positive on the inside of the cell now, millivolts. But that's all or nothing. It's like once you reach that threshold, you put your finger on the trigger and you just push enough, or put your hand on the toilet flusher, and you put enough pressure to reach the threshold to flush that water, it's not coming back. It's all or nothing. And that's what an action potential is. It's all or nothing conduction of electricity down a neuron or a muscle, right? So then it can't stay up there at this high voltage, because it would cause a problem with your muscles contracting, right? Or your nerves conducting. So we have to have potassium, which is normally inside the cell. It's an intracellular ion, which we learned before. Potassium will leave the cell. That's called efflux of potassium. So the sodium channels are closed at this point. So no more sodium can come in. So if you let more potassium out, that's going to repolarize the membrane back to resting membrane potential usually. because this is where we want to live with a resting nerve cell or a resting muscle cell. The muscle cell is probably just a little bit more negative, and you'll see. So when we talk about, first you need a stimulus. And in this case, the stimulus is the decision to move from your brain, right? Your cortex, cerebral cortex. That's going to stimulate this membrane of the neuron first to depolarize it. And depolarization is all about sodium influx. And if you reach a threshold, you're going to get action potential, which is all or nothing. Now that action potential can't stay up at that level. It has to repolarize back to resting membrane potential. So repolarization is all about potassium leaving the cell, efflux. Depolarization is all about sodium entering the cell. And that's what the signal is, from the neuron first, and then to the muscle. And then the muscle goes through the same thing. The muscle has a resting membrane potential. The muscle membrane or sarcolemma depolarizes. Then the muscle sarcolemma, the membrane, has an action potential. And then it will repolarize. And repolarization is about potassium efflux leaving the cell, making the inside more negative again. And that's the electrical part of this whole process. Okay? So, of course, you need ion channels, because this connection, like a connection between two neurons or a neuron and a muscle cell, like the neuromuscular junction, right? The neuromuscular junction basically forms a connection between the neuron and the muscle membrane. And in nervous tissue, whenever nervous tissue is involved and you have that kind of a connection, it's called a synapse. And this is a gap junction. They don't really connect. Sometimes the ion channels come together, but they really don't connect. So it's a gap with fluid in it. So synapse is a connection. And the gap junction technically is called the synaptic cleft. Okay, so you have ion channels, and what are the ion channels? You're going to talk about chemically gated channels, and there's voltage gated channels. So the chemical gated channels have to do with acetylcholine, and this is acetylcholine. That's the chemical neurotransmitter. Let me draw that word out. So right now, this is the only neurotransmitter you need to know, and you'll see where it's released from. So that's the chemical part. The voltage channels are all about sodium and potassium, mostly, right? And the voltage changes are depolarization and repolarization. So this synapse has two parts. It has a chemical part with a neurotransmitter, and then the voltage gated ion channels. The ions are sodium and potassium, right? We don't have to worry so much about calcium channels, because you do have to worry about it in the neuron. I shouldn't say that, because there are calcium channels that are really important in the neuron. But in the muscle, that the calcium channel, there is no calcium channels outside the cell. The calcium is stored and released in the sarcoplasmic reticulum. But in the neuron doesn't have a structure like the sarcoplasmic reticulum. The neuron has to rely on extracellular calcium to come into the cell in the neuron. And we're going to get to that. We're going to get to what it looks like, and then I'll go back and I'll put all of this together for you, because this is probably the hardest thing you'll do up until neuro, and this gives you a good background for when you do neurology, which is most of the semester after next week. So this is a channel on the muscle membrane. This is your muscle membrane. Remember the membrane? It's got biphospholipid. This is the sarcoplasm, and the fluid is the cytosol. and this is your extracellular fluid in the synapse, extracellular fluid. So and I can go into this. I'm just going to take it a step at a time though, because I don't want to do too much right now. So if this is the sarcolemma, right? Let me draw this better for you. Sarcolemma. That's this, right? This whole thing. This is a receptor. It's also an ion channel, right, on the membrane. So the acetylcholine, like this ball right here, is going to be released from the neuron terminal. It's going to be called an axon terminal or synaptic end bulb. It has different names. So the neurotransmitter acetylcholine will diffuse across the synaptic cleft and bind to a receptor, a specific receptor for acetylcholine. Now, this is a protein or a membrane protein. Now, this protein is going to allow, now when this acetylcholine never comes into the sarcoplasm, but sodium does. So when this neurotransmitter acetylcholine binds to its receptor, this allows sodium to come in great amounts, very fast. So because the sodium is coming in, it's depolarizing the membrane. And that's leading to an action potential if you reach threshold. So this is a voltage-gated ion channel. This could also be for sodium or calcium, and it could be and this could be the sarcolemma, or it could be the neuron plasma membrane. So this is not receiving a neurotransmitter. This is just based on maybe high levels of a certain ion, like let's say calcium. Calcium. So if this channel is open for calcium, calcium could come in, like say in a neuron. So let's do it this way. This is the muscle, right? Here, and this could be a neuron for calcium, but they both work the same as far as sodium channels and acetylcholine too. But let's just say this is the neuron. The membrane has a calcium channel. In this case, it happens to be open, probably because it was depolarized. So you don't need a neurotransmitter to open this channel. This one you do. That's why it's called a chemically gated ion channel. Like chemical is a ligand called acetylcholine in this case, a neurotransmitter. A neurotransmitter is the chemical. And there are other neurotransmitters, which you'll learn in neuro, like epinephrine or dopamine or serotonin or glutamate or GABA or glycine. But we're going to talk, all you need to know from muscle is acetylcholine. Now, this is a voltage-gated channel, and it could be for other. It doesn't have to be calcium. I'm just telling you that in a neuron, these voltage-gated channels are very important for calcium. Okay, so again, this is kind of throwing this at you in muscle when this is really neuro stuff. But this is the neuromuscular junction, how the nerve, the neuron and its axon are going to stimulate the sarcolemma, which is the muscle membrane. All right, so you really have to kind of know about a neuron. And the neuron is a nerve cell. Your brain cells are neurons, the nerves that move the muscles, or that excite the muscles, I should say, are neurons. It's really the only excitable cell in the nervous system. You're going to learn there's other cells in the nervous system called supportive cells, called glial cells. But for the most part, the nervous system is about neurons, and they have different parts. So the only neuron you're going to need to know in this for muscle, at the neuromuscular junction, of course, is a somatic motor neuron. Very important. Somatic means body, and somatic nervous system is all about moving skeletal muscle. That's it. Somatic. This is not autonomic, which is telling your heart to beat faster or your blood vessels to constrict. This is about skeletal muscle. It's not going to glands. It's not going, like I said, to your heart or your respiratory or digestive system. This is only going to skeletal muscle. And motor has to do with contraction, right? It's motor. And it's a neuron. It's a nerve cell. So the important parts of the neuron is really, in this case, is the axon, which is the part of the neuron that goes towards the skeletal muscle, right? And then each axon, of course, divides into many, many branches as it goes to different muscles, but we're only going to worry about one axon that reaches the neuromuscular junction. Sometimes they call that synapse. They call it the motor end plate. All means the same thing. So each fiber has one neuromuscular junction with a one motor neuron, but the neuron could have many branches. And one axon, depending, when you learn this in the nervous system, could innervate many muscle fibers. And some are just specific, like to blink your eyes, right? That's really a very high precision contraction and innervation. Where to contract your quadriceps or run, it's a lot different, right? It's a lot bigger, I should say, or less precise, right? I'm sorry, we got some background noise. Let me just, so again, the neuromuscular junction is what I was talking about before, that synaptic cleft between the membrane of the axon, really, of the neuron and the sarcolemma of the muscle. And we talked about high precision and low precision, and we'll talk about that later when we do the motor units and recruitment. So this is kind of showing you a nice picture of neuromuscular junctions. So this is kind of a good, simple way to show you this. Again, starts in the brain, cerebral cortex, primary motor area. It descends, because now this is descending from the brain down to the arm. Looks like she's looking at her cell phone to see what her grade was on that last take-home exam. And cell body of a neuron is really where the nucleus is. We're not so concerned about the cell body right now. We're concerned about the axon and its terminals. Okay, so now here's the, and they usually call this a nerve fiber. It's an axon of a somatic motor neuron. And this is somatic, not autonomic. And this is the synapse, or the neuromuscular junction. And you see that there's collateral branches for this axon. And these neuromuscular junction, this part of the axon, right? This part of the axon is called, it's got many names, but I think axon terminal is the best way to describe it at first. Sometimes I like to call it more physiological term like synaptic end bulb. Sometimes it's called Bruton, terminal Bruton. That's pretty much the three main, but I like to say synaptic end bulb or axon terminal. It means they all mean the same thing. Okay, so that's part of the neuron, and we're gonna have to look closer at the synaptic terminal, or that's another name, or the synaptic end bulb. So they call it axon terminal, which is easy, right? And I think that's the best way to learn it. And that's the end of the axon. So the direction of ionic flow, really, or action potential, goes from the brain to the neuron cell body, now to the axon, to the axon terminal. It's a one directional, coming from the brain to the muscle, to the neuromuscular junction. So there's a fluid-filled or gel-filled space called the synaptic cleft. Cleft is a space, and that's part of the gap junction between the neuron, the axon of the neuron, and the sarcolemma. So here's the key. Now in the axon terminals, and they look kind of like this, like say this is the end of the axon, here's the terminal, all that roundishness. So in the axon terminal, you have vesicles, which you remember from the cell, vesicles, right? And there's many in every axon terminal. So within those synaptic vesicles, you have neurotransmitter. And the neurotransmitter in the somatic motor neuron axon terminal in going towards the skeletal muscle is always acetylcholine. Okay. Now, on the muscle, you're going to see there's kind of like little folds in the membrane, kind of looks like this. And this is the muscle membrane, we'll say, or just a piece of it. This space is called the synaptic cleft. Okay, and this is the sarcolemma. You could say motor end plate, but I like to say sarcolemma. Let's call it what it is. It's the muscle membrane, which is very excitable. Okay. So this is the setup. Now nothing's happening here. There's no stimulus, but pay attention to this. When I use my brain to think about moving a muscle, that voluntary thought will depolarize the neurons of the brain, and a motor neuron of the brain. Eventually, it's going to come down to this axon. So this axon, I'll just say axon. Remember, it's an axon of a somatic motor neuron. It's been excited. It reached action potential. There's an action potential coming down here, right? Which means sodium's coming in like crazy. Sodium's coming in, right? So sodium comes in, that's depolarization, remember? And it's repolarizing as it comes down too, but now the sodium channels are open at this point. Now this is very important. I don't know what's going to say on the next page, but when that action potential reaches the synaptic terminal, the action potential is here where sodium continues to come in. All or nothing, right? It's not going to stop. It's going to reach here and then it's going to cause an effect. This is very important. When that action potential reaches the synaptic end bulb or the synaptic terminal or the synaptic end bulb, whatever you want to call it, it's the terminal of the axon. This will open voltage-gated calcium channels. Now, this is the neuron, remember. Calcium, when those calcium channels are open, calcium enters the cell and it binds to these vesicles. And when calcium binds to these vesicles in the synaptic end bulb, the neurotransmitter will be released into the synaptic cleft via exocytosis, into that watery or gel-filled synaptic cleft. So very important, and this is why calcium is so important for neurons. Remember, this is a neuron. This is the muscle down here. This is the cleft between them. This is the neuromuscular junction. So on the on the muscle membrane, on the sarcolemma, you have channel receptors. Draw them like this. You have receptors, and they're in the infolds too. You have receptors for acetylcholine, and they're protein channels that happen to be chemically ligated, which means it's the chemical part of the synapse. So acetylcholine never comes into the muscle sarcoplasm. It binds to the receptors. And once acetylcholine binds to these receptors, then sodium can come in because it's a sodium channel. It's not an acetylcholine channel. These are receptors. The acetylcholine stays in the synaptic cleft. It never enters the cell. So once you get sodium coming in great amounts, now you're going to depolarize the muscle membrane, and then you're going to get a muscle action potential. All right? So now, in the synaptic cleft, if you have too much acetylcholine in there, you can't keep acetylcholine, you can't keep any neurotransmitter in the synaptic cleft too long because that would be inappropriate. So these acetylcholine, or these molecules of acetylcholine, are broken down. So they leave the synaptic cleft. And that's an enzyme called acetylcholine acetylcholinesterase. Sometimes when you see ASE at the end, it's an enzyme. So acetylcholinesterase breaks down the acetylcholine that's left over in the synapse, because these are going to be saturated, these receptors. And you don't want to keep a muscle contracting, right? It has to stop. First of all, it has to repolarize. Second of all, it has to stop contracting. And this is one of the ways it stops. Acetylcholinesterase, which is an enzyme that destroys acetylcholine in the synaptic cleft. All right, so the enzyme is just reused over and over. That's never destroyed, but the acetylcholine is. So that helps stop the muscle contraction. So there's a lot of crazy stuff going on here, right? So again, to repeat, this is somatic muscle contraction. So you need the cerebral cortex. It's voluntary. You need a neuron action potential, a somatic motor neuron action potential. And that action potential will travel down the axon towards the synaptic axon terminal. And once that action potential reaches the axon terminal, the voltage-gated calcium channels will open and calcium will enter the axon terminal. Calcium will bind with these vesicles that are storing your neurotransmitter. In this case, in the muscle, it's always acetylcholine, which is always excitatory to muscle, somatic muscle contraction, skeletal muscle. So because of that calcium binding to those vesicles, the acetylcholine will flood the synaptic cleft. And then the acetylcholine will bind to its specific receptor on the muscle membrane or sarcolemma. And acetylcholine is the ligand. So these are ligand-gated channels, chemically ligand-gated channels. Well, acetylcholine is the ligand. The acetylcholine receptor is the receptor. So once acetylcholine binds to its receptor, now the sodium channels will open and you'll depolarize the sarcolemma, the muscle membrane, and you'll have an action potential along the muscle. Remember, the muscle fiber is a palindromic repeats of sarcomeres, right? And it'll continue down that. So now we're in the muscle now. Now, of course, you can't keep pushing acetylcholine towards the receptors again. They'll get saturated, but the help of this enzyme, well, it's kind of like Pac-Man. You know, Pac-Man, remember the game Pac-Man, where the big round head goes around eating up all the little dots and cherries and shit. So the acetylcholine esterase, sometimes we call it ACH, ACE, will eat up all the acetylcholine, so you can't keep getting bombarded with acetylcholine. And all neurotransmitters kind of work that way. Sometimes they're taken back into the synaptic end bulb. Sometimes they're destroyed by an enzyme like this one. So that's a lot of stuff. And we didn't even get to the actin and myosin yet, right? But now we're in the action potential. All right, so let me just get in your face for a second. Let's see, where are we? Let's end the show. See if you're awake. See if you're okay. Why isn't this stopping? There we go. Give you a couple seconds to turn on your cameras. You guys okay? So you're kind of following this. Now listen to this whole thing. We did a lot. This is one of the biggest things you'll do. So again, we're talking about skeletal muscle contraction. Skeletal muscle is voluntary. Know everything about the histology of a skeletal muscle cell. But because it's voluntary, we have to depolarize the upper motor neurons from our cerebral cortex and to send that signal of depolarization, action potential to the lower motor neuron. Now, this is all somatic. Somatic means skeletal muscle. So once that lower motor neuron is the somatic motor neuron, motor neuron is depolarized, reaches an action potential. That action potential is going to reach the synaptic end bulb. Now, once the action potential reaches the synaptic end bulb, it's going to create, or it's going to open the voltage-gated calcium channels on the synaptic end bulb. And when calcium enters the neuron, synaptic end bulb, calcium is going to fuse with the vesicles in the axon terminals that are storing the neurotransmitter. In this case, it's acetylcholine. Once calcium fuses with the vesicles, it's going to allow for exocytosis of the neurotransmitter into the synaptic cleft. So now we have flooded synaptic cleft with acetylcholine. Where does acetylcholine go? It binds to its receptor that's specific for acetylcholine. And the receptors are basically channels for sodium. So that allows sodium to flood the inside of the muscle, right? Inside of the muscle. Now, if sodium comes inside the muscle, it reverses the membrane potential. Now we have depolarization. And if we reach threshold, which I said was about minus 55, minus 65, it's more positive inside, we can reach an action potential. And if we reach the action potential, now the muscle membrane, the sarcolemma, is excited. And now the things start to happen. So what's the first thing you think is going to happen? This is a really hard question. Once the muscle membrane is excited, what's really the first thing that has to happen before we can even have that myosin pulling the actin towards the middle of the sarcomere? What has to happen? Anybody remember? It's a really hard question at this point. I'll tell you what happens first and see if you can tell me what happens next. So because the muscle membrane has reached an action potential, calcium is released from the sarcoplasmic reticulum. What does calcium bind to? What protein does calcium bind to first? Is it troponin, tropomyosin, actin, or myosin? Did I lose you?
Troponin.
Yeah, righto. Thanks, Jeremiah. The calcium binds to troponin. So because calcium binds to troponin, it could displace the tropomyosin that was blocking the binding sites for myosin on actin. So now we're ready to go. Now we can go into those 4 steps that I told you. Now, because that happened, because of this action potential, because of this release of ions, especially calcium from the sarcoplasmic reticulum, which bound to troponin, displaced the tropomyosin, now we can go to step 1, which is hydrolysis of ATP on the myosin head to energize it, to move it into position, to bind to actin. So that's the first step. The second step is the cross bridge, the connection of myosin to actin. Step 2. Step 3, with all that free energy that we got from hydrolyzing ATP, now the actin can be flung to the center of the sarcomere. The Z discs will come closer together, right? The H zone may disappear, the I bands get smaller, and the sarcomeres in unison will shorten, and that's muscle contraction. So now we have to stop it. So step 4 of this sliding filament configuration theory is detachment. It has to detach, right? And it needs ATP for that. You need to have ATP synthesized to make that detachment happen. So that's going to stop the muscle contraction. And don't forget, the muscle membrane has to repolarize as well, which repolarization is having potassium leave the cell, which makes the outside more positive and the inside more negative again, back to resting membrane potential. So that's stopping the muscle contraction as well. The third way the muscle contraction stops is all that acetylcholinesterase, that enzyme that destroys acetylcholine. It's in the synaptic cleft, will destroy the acetylcholine and stop the possibility of more contraction that's inappropriate by destroying the acetylcholine. And that's how it stops, right? So let me just go a little further before we take a break here. Where were we anyway? Where were we? I was in a slideshow. We're almost done with this one. Oh, this is the wrong PowerPoint. That's why. Let's share this with you. We'll take a break in a minute. Are we still recording? Yes, right? Perfect. Okay, so this is a close-up of the axon terminal. This is a really good picture, actually. And you can see the vesicles. You can see some mitochondria in there because you need to make ATP for all this to happen, even in the neuron. So the yellow is all the neuron. It's the motor neuron. This is the axon, and this is your axon terminal, this bulb. That's why they call it a bulb. So again, it's not showing you, but this is the calcium channel. So calcium has to come in after you have an action potential, of course, down this way. It's one direction. Calcium enters the cell, fuses with the vesicles, and then exocytosis of the neurotransmitter. Here are your acetylcholine receptors that are actually sodium channels to allow sodium to come in. And once sodium comes into the cell, it depolarizes it, which makes a lot of sense at this point. Okay, so again, to repeat the events of the neuromuscular junction, the big picture, which I just pretty much told you, it's just not easy to get all the parts together. All right, you need a muscle fiber excitation, and that comes from the acetylcholine binding. Really, that's the chemical part of this process. And there's excitation, of course, because acetylcholine is always excitatory to skeletal muscle. And then you have your cross-bridge cycling, which has to end in detachment. Okay, so this is pretty much the events to the cross-bridge. But again, you have to have power stroke, right? After this would be the power stroke. And then detachment. And then the muscle can relax. So you have to kind of put this together and listen to the recording when we're getting ready for this test. So this is at the neuromuscular junction, the action potential, but you have to know how it got there, right, from the cerebral cortex. And this is really, they didn't show you a picture of this yet, but this is a key part. When voltage-gated calcium channels open in the neuron, this is the synaptic end bulb, not in the muscle. So the acetylcholine diffuses into the synaptic cleft through exocytosis and diffuses to its receptor. In this case, it's ACH, acetylcholine. And then your sodium gates open, and you have end plate potential, which is depolarization. And then you have action potential. Okay? And then how it stops is the calcium is sent back into the sarcoplasmic reticulum, and acetylcholinesterase will degrade the acetylcholine. All right, so put all this together, right? And here's more of the same. At least now they're showing you the calcium channels, right, on the neuron, right, in the axon terminal. Then you have your ACH receptors. And this is just a close-up of the neuromuscular junction. See, it's a gap junction called a synapse. So the action potential has to arrive. The sodium channels open on the synaptic terminal. Calcium entry causes the exocytosis from the vesicles, and then diffusion of acetylcholine across the synaptic cleft, that binding with the acetylcholine receptors specific for acetylcholine. And that opens up your sodium channels for sodium to come in. More important than potassium leaving at this point. It will repolarize later. It happens very quickly, though. It's very quick. Sodium channels are very fast. Fast sodium channels. They open up and allow sodium basically to flood into the cell. So then the acetylcholine is destroyed by the acetylcholinesterase. It's degraded, they say. Acetylcholinesterase is the enzyme that is specifically breaking down acetylcholine. So a little film here. You watch a little AP flicks. Instead of Netflix later on, you watch the AP flicks. Okay. So any questions there? PowerPoint is mostly about the action potential, some review. So let's go through the chemical and the electrical part of this excitation, ultimately of the neuromuscular junction of the sarcolemma of the muscle to cause contraction. Again, you understand the whole idea of the sarcomere. This is just mentioning some things. We're going to talk more about musculoskeletal conditions in the next PowerPoint and some clinical applications, but I just have to get you through this electrical part especially, because I think that's the hardest thing. So we're talking about polarization, right? Where you have, I'm just going to draw a quick membrane. Of course, you know it's biphospholipid. This is the sarcoplasm on this side, which is the inside of the cell, or intracellular fluid. Then you have the ECF, which could be anywhere. It could be anywhere along the cell body of the neuron. It could be along the axon of the neuron. It could be along the sarcolemma. So the inside at rest, in the resting membrane potential, is very polarized, means very positive. Oh, sorry, it's negative on the inside, sorry. Very negative on the inside and very positive on the outside. All right, and that's the resting membrane potential. So the very negative, like minus 70 to minus 90 millivolts inside the cell. We don't really measure the outside of the cell. It's a vast area, right? And the membrane has proteins, like could be channels, voltage-gated channels. You could have receptor proteins that are also part of a channel, and they're proteins along that biphospholipid membrane. So this is your resting membrane potential, and we call that the RMP. So it's not exciting at this point, but it's excitable, and that's where we want it to be. We want to always maintain that resting membrane potential between action potentials. So depolarization is sodium coming in. And remember, sodium is positive, right? It's a positive. It's a positive ion, a cation. And that's more abundant on the outside of the cell in the resting potential. That's probably why it's so positive. The inside has potassium. Potassium's abundance is mostly on the inside of the cell, but I know it's positive, it's a cation. But the inside of the cell is more negative because it has very large proteins inside that carry a negative charge. Plus you have phosphates that also carry negative charges. So this is why it's so negative inside. The potassium is meaningless as far as the potential and polarity. So sodium influx will change the membrane potential and cause depolarization. So it started polarized, now we depolarized it when sodium comes in, right? And then ultimately, the sodium channels have to close. Those voltage-gated channels will close, and so sodium stops coming in, and then the potassium channels may open, and potassium leaves, and that makes it even more positive outside. So repolarization of that membrane, back to resting membrane potential, is more about potassium efflux. Sometimes excess potassium comes out, or sometimes, I just want to add this right now, it's not going to affect the muscle contraction so much, but the chloride is a negative ion, an anion, and let's say chloride comes in during a resting membrane potential, that's going to hyperpolarize the membrane, right? Because that's going to add more negativity to the inside. So I could add to this a condition called hyperpolarization. Which would happen if, say, you have more potassium efflux Or you have chloride influx, because that's negative, just so you understand the polarity that's going on here and how this is going to be affecting the neurons of the nervous system, which we're going later in the semester. So now this is the chemical part now. Chemical part, the acetylcholine. And you should know now how it's released from the motor end plate, from the synaptic end bulb. And that's the synaptic end bulb of the motor neuron, or axon terminal. So the acetylcholine receptors are on the sarcolemma, and now they call these chemically gated ion channels, or ligand gated ion channels. They're going to allow sodium to come in to further depolarize. So sodium diffuses into the muscle cell. But potassium doesn't make a difference, even though the sodium and potassium may share that same carrier, or same channel, I should say. It's not a carrier, it's a channel. So the exiting of potassium at that point makes no difference, because sodium is diffusing in massively and very quickly. So the inside becomes more positive, right? And it results in depolarization of the end plate. Now, of course, that threshold has to be met, right? So that threshold, if it's met, will cause or produce action potential in the muscle now. The neuron already met its action potential. So you have depolarization of the muscle, and that's your end plate potential. End plate is the muscle, the sarcolemma. So here it is. So once this acetylcholine binds to its receptor and the sodium comes in, then you have depolarization, a wave of depolarization along the whole membrane. It keeps going from sarcomere to sarcomere, right, along those ion channels within those tubules. And of course, calcium is very important from the sarcoplasmic reticulum. So depolarization will propagate an action potential. An action potential is all or nothing, and it has to repolarize. So if there's enough voltage, enough sodium coming in, enough change from a negative number to that threshold, then the sodium channels will continue to open. And that's why they're voltage gated channels. So large and very fast influx of sodium triggers the action potential, which is all or nothing, unstoppable, like flushing a toilet. And that action potential will lead to the release of calcium from the sarcoplasmic reticulum. And as the action potential spreads from section to section of the muscle membranes, muscle cell membranes, it will continue to have the muscle contract until it repolarizes, or the acetylcholine is gone from the acetylcholinesterase. So this is again showing what happens during action potential. The sodium channels are open, right? Potassium channels are closed, waiting to repolarize later. So remember, sodium in is depolarization. Potassium out is repolarization when the sodium channels close. So now the sodium in repolarization, which has to happen after an action potential, right? And that's what it says up here, action potential. I know you can't see it there. Let me just get this out of the way. The sodium channels close, and then potassium gates open, and then you have potassium efflux, which makes the outside become more positive again, because the sodium is already there. No matter how much sodium comes in, sodium is always high in the extracellular fluid. It's a very, very abundant cation. Now, this is an important part. This is a refractory period, and it's usually just as the membrane is finishing repolarization. So this means a refractory period, this is kind of important, a refractory period during this depolarization, action potential repolarization, is a time during that change, during that polarity change, where no matter what happens, if you send another stimulus, if you get a very strong stimulus, you're not going to get another action potential until repolarization completes back to RMP. So refractory stops another quick action potential that could be inappropriate. Very important in heart muscle, right? You want that to be completely rhythmic. I mean, there's absolute refractory and there's relative refractory. Like sometimes absolute refractory is when there's no way, no how you're going to get another action potential during this period, right before repolarization is complete to resting membrane potential. So that's really all we need to know about refractory period right now. Again, that's more important in cardiac muscle. So now we have the condition where we want to stay at the resting membrane potential. So these installation of these pumps, sodium potassium pumps, are really important to maintain the resting membrane potential. So really, technically, they are called your sodium potassium ATP ACE pumps. And these are basically an enzyme. This is an enzyme that's going to cleave ATP. What ATPase does is hydrolyzes ATP, like I told you before with the myosin heads. So ATPase added to ATP and plus water will cause ATP to go to ADP, adenosine triphosphate to adenosine diphosphate, plus that high power inorganic, that's the I is inorganic phosphate. And that's what really gives energy. Now, why do we need ATPase for these pumps? I'll tell you why. Because here's the membrane, right? And here's your sodium potassium ATPase pump, which is a protein embedded in the membrane. Here's the ECF. And here is the cytoplasm, or the intracellular fluid. We'll call it sarcoplasm, why not? Because we're talking about muscle. So sarco means muscle. So remember, there's more sodium always outside the cell. A lot of sodium outside the cell in the ECF. And there's more potassium inside the cell, normally. So now we have to move sodium out of the cell, right? And that's going to have to go against its concentration gradient. So it's going uphill. It's active transport. This is not diffusion going from high to low concentration. This is low to high. So you need energy. At the same token, you're moving potassium back into the cell. And that takes energy because you're going against the concentration gradient of potassium. So you need energy to do that. And that's why this is ATPase. This protein is ATPase. So it's constantly pumping 3 sodium out and 2 potassium in. Always, all along membrane. And pretty much every cell has this, especially these excitable membranes. And what these pumps do, the sodium potassium pumps, ATPase pumps, is maintain that resting membrane potential, keeping it positive on the outside, right? And negative on the inside. It's the most important way to maintain the resting membrane potential. Because now we're at a refractory, we can get another clean action potential. That's the best way to describe it. So more about the chemical and the channels and how it closes. So this is your chemical part with the acetylcholine binding, which opens up the sodium channels. And that's depolarization right here. Potassium channels are closed during depolarization. Then after the action potential, we have to repolarize. And there's a slight period of refractory during the end of repolarization. So now the sodium channels will close and the potassium channels will open, allowing potassium to exit. So you look at the membrane negative and positive during all of this. It gets more positive on the inside, more positive, and less positive on the outside at these segments. Because it's really where we're talking about is right up against the membrane, right up against those phosphate heads, right? So remember, this is the ECF out here. And this would be the sarcoplasm in all the pictures. So repolarization now, it goes back to positive on the outside in the ECF and negative on the inside. So we have a nice resting membrane potential ready for another round of depolarization if we need it. And it's optimal for muscle contraction, especially somatic muscle contraction of skeletal muscle, voluntary. So this is something you should remember. This is the graph of what's going on with depolarization and repolarization action potential. So the membrane potential is on this y-axis over here in millivolts. So here's your millivolts on the left side, and the bottom is time, which is irrelevant right now. You don't have to worry so much about the time. It just takes a few milliseconds for all this to happen. And it's measurable too. So you're in a muscle cell, it's very negative. So the neuron is minus 70, but in a muscle cell, this is about a muscle cell now, the resting membrane potential is minus 90, right? So that's the resting membrane potential inside the cell is minus 90. Now, when the sodium channel is open because of a stimulus, like me thinking about moving my muscle, or a neuron with an action potential is stimulating the sarcolemma, and we're talking about the sarcolemma. So the sodium channel is open, and that's depolarization. So now we're getting less negative as you're going up. And if you reach a threshold, let's say minus 65 millivolts, that would be your threshold for action potential. So once this depolarization gets up to minus 65, then it's all or nothing. It continues to depolarize and hits the top, and then you have an action potential. This is the peak of action potential, which in this case is 30 plus, or plus 30 millivolts action potential. Now it's all or nothing, and it has to repolarize. So now on repolarization, sodium channels close and potassium channels open. Now you can start repolarization, going back down to negative numbers. And once it reaches that resting membrane potential of minus 90, then the potassium channels can close. Now say the potassium channels, or the potassium comes in too much, and you come down here and then go back up here. So this would be, say, minus 110, which is very negative, millivolts, would be hyperpolarization. And once you get into hyperpolarization, it makes it very difficult to get an action potential. Sometimes that's completely appropriate in other neurons, but we're talking about the muscle cell, which we want excitement here. But we also want repolarization. So usually around here, like at the end of repolarization, back to resting membrane potential, like between here, you have a period where you can't, even if you get a really good stimulus, you can't get another action potential at this point. You could definitely get one. Sometimes you can get one here and go up, but here you can't, and that's called your refractory period, where you can't get another action potential. Okay? So this whole thing is basically called excitation-contraction coupling, where the neuron excites the muscle membrane, which is the sarcolemma. And then that excitation, the action potential, AP, continues along the muscle membrane from sarcomere to sarcomere, creating release of calcium, right? And the release of calcium, and where's the calcium stored? You have to know this. It's in the sarcoplasmic reticulum. And calcium, like Jeremiah says, in the muscle is going to bind to troponin. And then troponin is going to displace the tropomyosin off of the binding sites for myosin on actin. And then we could hydrolyze the myosin heads with ATP, ACE. And then you could form the cross bridge. Then you could have the power stroke. And then you could have detachment with more ATP. So action potentials are fast and short-lived, but they're all or nothing. Just like flushing the toilet with enough pressure to get that toggle switch or the flusher handle to create that flush, right? So any questions about the action potential? And we'll go on to more metabolism. I don't know if we'll get through this, but we'll do as much as we can if you guys hang in there for a bit. So let's go over to muscle metabolism. This is a little less intense. I think I'll go as slow as I can. We'll go through here so I can see this. So this might have a little bit more action potential, but I don't think so. So this is just talking about the type of muscle contractions we have, like gross anatomy muscle contraction, like whole muscle, really we're talking about whole muscle, which is a combination of single fibers together. So contraction, the shortening of the sarcomeres creates a tension that really does the work. Tension is a force on a load or an object. So you might have heard of it like isometric contraction, where there's really no shortening of the muscle. So the tension's increasing because you're putting a load onto the muscle, but there's no movement of the joint, right? So it's kind of equalized pressure, like if you were standing up and just bent your arm and held a book in one position. It didn't go down, it didn't go up, right? There's no lengthening, there's no shortening either. Shortening. Isotonic now has movement of the joint. So like tonic means movement. So isometric is tension on the muscle without movement or shortening. So isotonic has shortening, and it could have lengthening too, right? Because the tension exceeds the load. Like if I'm holding the book out isometrically, and then I want to bring the book towards my shoulder by contracting my biceps brachii, now the muscle tension is going to exceed the load, so I can shorten the muscle to do that work of flexing my elbow. So think tonic is movement, isotonic. Isometric is no movement, just a stability or a static position. All right. So talking about motor units now. So each muscle is going to be served by one of the motor nerves, which is a motor neuron or an axon of a motor neuron. So a motor nerve now, again, has multiple axons, multiple neurons. A nerve is a group of neurons. That's what a nerve is. Like you can't really call an axon a nerve. It's a fiber. It's just a neuron. But a nerve is bundles of axons. So you could have hundreds of motor neurons in one nerve. It depends on the nerve, and you'll learn the nerves. You learn the brachial plexus and the lumbosacral plexus with the sciatic nerve and all those. So the axons will branch to the neuromuscular junctions, right? And usually sometimes within a single muscle fiber. It depends on the precision. I was talking about that before, like this. Like high precision is your eye muscles, movements of your eyes or your eyelids even. But low precision would be like your quadriceps, which has many, many neuromuscular junctions, many motor units. So the motor unit is the muscle that's innervated by one nerve. So one nerve, like say an L4 nerve coming out between L4 and L5 lumbar, or L3 and L4 lumbar, will serve a certain amount of quadriceps muscles. So one motor unit is the one nerve and all the muscles that it innervates. Innervates means to give the depolarization, to give the power for the muscles to contract, so to speak. It's not really power, it's electricity. So yeah, I guess you could say power. But I don't want to confuse you with the actual powerful muscles to do work. So here's a better definition. A motor unit consists of the motor neuron, all the muscle fibers from that motor neuron that it's innervating. So like this is what I'm talking about, high precision. The smaller the number of fibers, like in your eye muscles, the greater the fine control or high precision. All right. So again, you're going to need a lot of motor units in your someplace like your quadriceps, because if they're not all working, you're going to have weakness in that muscle and a weaker contraction. You're not going to get a full contraction. So this is showing you two different motor units in this case. One motor unit and two motor units, and I don't know what muscle this is, but it's a pretty long bone. This could be a biceps brachii or a quadriceps, but it looks more like an arm muscle. So this is showing all of the collateral branches with their axon terminals come into contact with multiple muscle fibers. Like here, like this motor unit is affecting here, and this motor unit is affecting here. And this is the spinal cord right here, which again you'll learn later. And motor neurons are coming out this way. And this is a nerve here, which is a bundle of many axons of neurons. So that's why one nerve could affect multiple motor units. It's hard to explain that before you learn the actual nervous system. So this is, it gets a little complicated, but this is what I've been talking about most of the time, a single muscle twitch, where you're just going to have a quick, and this is a different graph, but you're going to have a quick depolarization, action potential, and repolarization. And you get one stimulus at a time. So, the left axis, the y-axis would be the strength, the tension. And over here is still time. So again, this is not action potential, but it kind of fits nicely into the action potential graph. But really talking about the strength of the contraction. You start out no tension, and then it goes up, and then comes down. So it contracts and relaxes all in the same with one stimulus. Okay, and a myogram is actually a muscle test, like an electromyogram. Will trace the tension. It's really checking the tension, not the millivoltage like we saw before. So this is a different graph, so hopefully we'll see one coming up soon. So there's a latent period, of course, like if I'm thinking, right, if I'm thinking about, and this is muscle tension, not millivolts, if I'm thinking about, I think, like right here, I think about contracting my fingers to write. And then I contract, then it contracts, then it relaxes. So that's one muscle twitch. So the time it takes for me, that thought to go from my cerebral cortex to my forearm muscles and my finger muscles is called the latent period. Right? So there's no contraction, and it's milliseconds, right? It's myelinated fibers, which you'll see are very fast. But still, there is a latent period where it takes time, a split second, to get the muscle to actually contract. And this is the contraction, maximum part of the contraction. So this is the period of contraction here. And then you have the period of relaxation as the muscle relaxes. And again, this is not the same chart. It looks the same, but this is a muscle twitch with tension being over here on the left axis, on the y-axis. It's tension, not millivolts. So this is interesting because it tells you what happens. So in the latent period, you're thinking about moving the muscle. That was your thought. That's the stimulus from your brain. So it takes a few milliseconds to get there. And the further away it is, the longer it takes, believe it or not. And then you have the contraction is when you actually have that cross bridge between actin and myosin that's shortening the sarcomere after the power stroke. So as that's moving to the center of the sarcomere, Z discs coming together, the tension increases. That's why the tension's here is at its peak. Right? And then to relax a muscle, the calcium is sequestered or re-enters back into the sarcoplasmic reticulum. And then the muscle can't contract anymore, so it relaxes. So this is a single muscle twitch with one stimulus. What's the stimulus? Me thinking about contracting a muscle because it's voluntary skeletal muscle. So this kind of shows you what goes on here. So that, again, thinking about it, you have your latent period. So you have zero tension, right? This is percent of tension. And now, once that action potential reaches the muscle, then tension increases and you have the period of contraction. And then you have a downward slope. So this is not so steep in a muscle twitch. Period of relaxation takes a lot more time than it did to get the contraction to happen. The duration of contraction is a lot less than the period of relaxation, which is interesting. And this is time in milliseconds. So the latent period looks like it's, maybe 2.5 milliseconds for that thought to get to my muscle action potential. Right? And these are isometric twitches, just contraction of the muscle without movement of a joint. That's all that means. So the strength and duration of the twitches varies to what's going on in the muscle, really. Like what, and we're going to talk about muscle metabolism. Hopefully we'll get to some of that tonight. So there are different muscle fibers when it comes to metabolism, which you'll see. So eye muscle contractions are brief, right? And probably more fatigable if you keep contracting them. Like if you keep moving your eyes, they'll fatigue rather quickly. Where the thicker muscles will hold the contraction more continuously. So the time of contraction, again, and this is a muscle twitch though. This is not like postural, holding your muscles contracted for long periods of time. In fact, this is less than a second of contraction. This is a muscle twitch, quick contraction. And I hope that's understandable. It's just a quick contraction relaxation. There's no sustained contraction is the best way to describe that. So this is pretty cool because this shows you the different muscles that have longer periods of contraction for just a muscle twitch. Like your eye muscle, this is an eye muscle, lateral rectus, which moves your eye to the side. So the latent period is the same for all of these. And the lateral rectus has a very short period of contraction. So it probably doesn't have a lot of ATP to spend. But gastrocnemius, which is much more external muscle you're going to learn in the back of the leg, in the calf muscle, the two big headed gastrocnemius, has a little bit longer than the lateral rectus, but it's not that long of a contraction period and more steep of a relaxation period. Soleus is deeper muscle in your calf. It's like right against your tibia. So this is built and has different metabolism. So if you look at this one, this has a much slower and wider duration of contraction. And this is just a muscle twitch. And this is just a latent period of contraction, and then period of relaxation. No sustained. Like a sustained contraction would keep going up here and go on for time. Think about that. When you're standing up for a long period of time, how your muscles need to continually be stimulated and contract. But this is not what we're talking about. This is a laboratory with a myogram just showing you a single muscle twitch in these three different muscles. Lateral rectus, high precision, very small muscle. Gastrocnemius is a pretty big, thick muscle on the outside that points your toes down, called plantar flexion. Soleus basically does the same thing, but it's a different metabolism. It's deeper muscle. So you could change the muscle responses, like different demands, like if you want to do a quick move or you want to do a sustained contraction, and your muscles have different types of fibers, actually, different types of muscle fibers that some of them are built more for like sprinting. Some of them are built more for marathon running. Big difference. Some of them are for powerlifting, while the others are for more triathlons. So again, and then there's the non-athlete who just, you just need to keep your posture, or you just need to pick up your books off the ground. There's different demands for muscle to do work. And, it depends on how much you're stimulating those muscles to do work, whether it's short period of time, like a muscle twitch, or a longer sustained contraction. like standing up for a long time, or even just looking at a computer for a long time, or working on a computer for a long time. So muscle twitch is a single stimulus. That's the point here. That's important. Right, so here's your muscle twitch with 2 stimuli. And when you have multiple stimuli like this, of 2 stimuli, they call that summation, and temporal versus see, spatial summation is the other summation. So here's your stimulus. This is the motor neuron stimulating the muscle to contract and relax. And then there's another stimulus within a relatively short period of time, maybe 100 milliseconds between each stimulus or so. So the complete relaxation was allowed to happen till you got the next stimulus. So these are 2 muscle twitches, just separated by temporal summation. So then you have multiple stimuluses, but you're going to get more of a wave summation. So now this will never get a chance to relax totally, and you're going to see that. So they really don't have time because a new stimulus came in. And this is what happens with postural muscles, right? And each stimulus increases the force. And this is all about balance and keeping you upright, you know. So of course you need more calcium released from the sarcoplasmic reticulum every time that stimulus hits that muscle from a neuron. So this, again, look at the multiple stimuli, the red arrows, or how many times this muscle is going to be stimulated. So it's multiple stimulation called temporal summation. And because this is going up, like here, it never had a chance to relax, and boom, another stimulus came. Kept going up, it got stronger, another stimulus, and stronger. And then it gets to a point where it just gets high, and basically you hit fatigue. So fatigue is like a forced relaxation, because after a while you run out of ATP and oxygen, and calcium gets sequestered back into the sarcoplasmic reticulum. So this is temporal summation, also called wave summation. So you're getting multiple stimuli, and each time you get that stimuli, there's a stronger tension in the muscle. But it can only go so far before there's fatigue. You run out of ATP, you run out of oxygen. And you can always pay it back later by resting, right? So now we have major stimuli to the point where you're going to get beyond the waves and get a fused or unfused tetanus. So that's an incomplete tetanus. Now, tetanus is a constant, many stimulations. It's not just the stimulations, like you're getting constant stimulation to a muscle where it goes up, and then it just kind of plateaus, and that's tetanus, which is completely appropriate. I'm not talking about a tetanus bacterial infection. I'm talking about a sustained, appropriate contraction that's going to do work to hold you upright for longer periods of time. And you need that sometimes. So unfused tetanus is when you still got the waves, but it kind of plateaus. You know, you see it's going up and down within that same tension. And then, of course, it's going to fatigue. So you have even more stimuli, and the stimuli can get faster and cause that high tension wave. So this is unfused tetanus. So another difference between a muscle twitch, temporal or wave summation, and now unfused tetanus, because you have that up and down. The unfused part is this trough peak, but still it's getting multiple stimuli over time. So the time is a little more important. Now, fused tetanus is going to be a flatter top, which you're going to see. See the contractions fuse because you're getting even more stimuli. Now, of course, if this continues, it can't stay like that forever, of course. And these are skeletal muscles, right? So you know what that feels like when your muscles are contracting for long periods of time. You will get muscle fatigue because you're going to consume all the oxygen. You're going to consume all the ATP. So the energy is low and the fatigue sets in. So now you have massive, very close together just enough to get out of the refractory period of stimuli, action potentials. And these are action potentials. Let's call it what it is. So this is fused tetanus. So you have, and it's very smooth. This is completely appropriate to have this going on for periods of time. And the better condition your muscles are in, and the less fatigable the muscles are, you'll be able to hold this for longer periods of time. But again, ultimately, it's going to fatigue. Right, fatigue. But again, you pay this back. You'll rest. You'll get the oxygen in. You might have to breathe a little faster, harder, deeper. The oxygen gets to your muscle tissue, and you start storing more myoglobin and more glycogen and making more ATP again, storing that energy for the next contraction when necessary. In every way, not just athletes, of course. So muscle responds to changes in strength. Like sometimes you have to recruit motor units. All right. Sometimes you're doing an exercise where most of the job is being done by your quadriceps, but sometimes you need your gluteus maximus to help out with that movement, because the quadriceps might get fatigued a little bit. It's not a very fatigable area, your quadriceps, normally, but then some other muscles have to be recruited. Right? So sub-threshold stimulus is not going to get an action potential, so you have no contractions. Right? If not enough depolarizations get into that muscle. So a threshold, just at threshold, is strong enough to cause an action potential, but just might be a single muscle twitch. Maximum stimulus, strong stimulus, and then that increases the maximum contractile force, and it recruits other motor units to do more work. Like if you're just getting up out of a chair, you're not going to have to use every motor unit to get up, so you don't need that much stimulus. But if, say you're doing a squat, you're putting weight on your back and you're doing squats in the gym, or you're going from a squat position up to upright, and you're increasing the load, and therefore you have to recruit other muscles to contract to do that work of that squatting exercise. So again, this is showing just a relationship, and this is really not that important. Are you ready? Just know the summations and the muscle twitch. So again, this is just showing your stimuli getting stronger and stronger and stronger, right? But it depends on the threshold. So if you meet the threshold, the maximum contraction can only contract so much. And plus you can only get actin and myosin to slide to the M line. You can't get more contraction than that. There's an end point, which is always important, like the maximum physiological contraction you can get. And the same thing with stretch. If you stretch a muscle, you can only stretch it so far before it's going to tear, right? So you need optimal lengths between these actin and myosin in these sarcomeres. So it's really important. These are important facts about the size and strength. Motor units with the smallest muscle fibers are recruited first, and then the larger fibers. And of course, the largest motor units are going to be the most powerful. All right, and you're going to see like there's different types of fibers in large muscle groups. Mostly talking about large muscle groups, like your back muscles, your quadriceps, your hamstrings, your calves too, your pectoralis, right, which is your chest muscles, which you'll learn all these muscles, believe me. It's not as bad as bones, but it's pretty close. Okay, so just looking at the size and the time of contraction here, this is really not that important that you look at this graph and figure it out. The other two are much, much more important. Sorry. Okay, we'll talk about spinal reflexes. We're gonna have to talk about that in neuro because it's a whole nother story. And it involves reflexes or skeletal muscle contraction, somatic especially, that don't involve your cerebral cortex. So you can, when you hit somebody in the patellar ligament with a hammer, you're testing the lumbar reflexes of the quadricep muscle. So the quadricep will contract when you tap somebody below the kneecap on the patellar ligament, but you don't need your brain to contract that quadricep. So that was a reflex, only involved the spinal cord. So again, this keeps a firm tension in your muscles to keep your balance, to keep your posture good. And that's muscle tone. Sorry, that's muscle tone. So the tone is the reflexive contraction of muscles to maintain your integrity of your posture, right? And keep you balanced, keep you stable. And of course, that needs a bit of resistance too. So isotonic, we mentioned this before, where you have movement of the load and change in length of the muscle. Like I was telling you, when you hold a book out straight, it's isometric. But if you move the book towards your shoulder, that's isotonic, and that's concentric. So the biceps muscle is shortening as you're doing bicep curls, right? You bring your book or the weight dumbbell towards your shoulder. You're flexing the elbow flexion, and that's shortening the biceps muscle, biceps brachii, and that's concentric isotonic, because isotonic is movement, isometric is not movement. Eccentric is lengthening the muscle as it's contracting. So if you're holding the book or the dumbbell isometrically in the middle, and then you let it down slowly towards your thigh, that's an elongation of the biceps brachii, and that's an eccentric contraction. So eccentric is lengthening the agonist muscle, the muscle that's doing the work. And of course, there's reciprocal contraction. Your triceps muscle is also doing the opposite, like an antagonistic movement, which you'll learn. Okay. So here's the isotonic concentric, where the muscle shortens to lift the load. That's an isotonic concentric. So you might see this actually on a lab practical. Sometimes this shows up. Difference between isotonic concentric and eccentric. So again, this is concentric again, talking about the length is decreasing, dropping as the tension increases. That's isotonic concentric. Isometric, there's really no movement, right? There's no movement. You're just holding both, so the load is the same. Some more isotonic contractions. The actin filaments are getting closer to the middle, causing shortening. Isometric, there's a force, but the actin does not shorten. So again, it's still doing work, though, to hold that book or that dumbbell straight out in front of you with your elbow flexed. So this is isometric. There's no change in length. There's no change. Oh, but you're still holding that weight up. So the load is not exceeding the tension. Right? So there's no change. There's no change at all right in here. Somebody have a question? Question from anybody? Okay, so now we'll talk about the energy needed for these contractions. Of course, ATP, right? This is our energy source for all our cells. So of course you need ATP to be cleaved and hydrolyzed just to get the myosin to move. And that extra energy will cause the movement of the myosin head to the actin. And then you need ATP to detach the cross bridge. And then you have to, these pumps need ATP as well to get calcium back into the sarcoplasmic reticulum to stop the contraction. And you have your sodium, potassium, ATPase pumps. So it's a lot of ATP involved in muscle contraction. So the available ATP stores are depleted in 4 to 6 seconds. So again, I think we're going to do this next time, because this is very interesting, but it's going to take a little, we really have to go through that.